Hello everyone. Today I'm going to talk about the Connectome, synaptome, and Alzheimer's disease. I'm going to start with the introduction. Then I will talk about the promising technologies to study the microorganization of the brain. And then I will talk about the idea of the neuronal excitation inhibition balance as the endpoint of all Alzheimer's disease-related micro-level synaptic pathology. So when we see a picture like this, begins our fantastic journey into the brain, starting with the retina, and then with the thalamus, then the flow of information start to travel through these very dense neural forest that constitute our brain with more than 100,000 million neurons. So we see one of the questions that we have here. Well, okay. So one of the problems that it is a mystery. What is how you can relate the brain activity of the circuits with cognition and behavior? We don't know. We don't have any idea of how this activity gives rise to behavior and cognition. So, but one of the first steps toward understanding how neural circuits contribute to the functional organization of the brain is to get the detailed connectivity pattern of the brain. And we will see that there are three major distinct levels of analysis of the connectome, the synaptome. The one is the macroscopically, major tract connectivity in terms of imaging, like they have several people have been talking about. Then we have the intermediate resolution, that is the light microscopy, where you can map the putative synapses. And then we have the ultrastructural level map of synapses, which is the most difficult because it's a very nano scale. So we will see that now we have very fantastic methods to analyze the human brain. Instead to use data from an experimental animal, we can use directly from the human brain. So the brain, the cerebral cortex, is considered to be made of microcircuits that are in all the cortical areas. There are vertical connections, also horizontal connections, and the afferents from the external world rise to layer 4 and upper layers. At the end, the end product of this activity is the interaction between the memory, the external information, and the one that is generated by the own brain. It's an unconscious activity. We don't know what is why we have this kind of activity, but this also interacts with the column. So the pyramidal neurons are responsible of the spread of the activity through the axons collaterals and are the major source of excitatory connection. These are a very important concept. So all these activities of the brain has to go through the action of pyramidal neurons. So the pyramidal neurons, we will see in the next slides, is a major it's the main type of cortical neuron that represent about 70–80% of all cortical neurons. They are the major source of excitatory synapse, and the spines, or the dendritic spines, are the major target of this excitatory input. So the vast majority of connections, excitatory connections, arise from pyramidal neurons, and the spines are the main target of excitatory connections. So understanding the principles that determine the dendritic geometry is critical to construct then in silico digital circuits in the brain. So we need to know what is the design of the neurons that make up these circuits. So in analogy to the electronic circuits, neurons correspond to electronic paths, and neural cell types correspond to what kind of paths they are. And this is very important because the classification of cells is not only important from the I have to get my computer because I have a lot of movies and then so So the point is that you can get information that before was not available about all the details about the postsynaptic densities, the postsynaptic elements in the human brain. So the idea is now to go to use the same computer reconstruction that we made with Henry Markman in the paper that we published in Cell, where we include the neurophysiological and anatomical data, physiology, synaptic levels, and to integrate this information, like in the instead to be a rat brain, which is a draft. Now we are trying to incorporate all of this data and make this in silico models. So now talking about Alzheimer, Alzheimer's disease is the most common form of senile dementia, and the main pathological hallmarks are the presence of extracellular beta amyloid that is deposited in the brain, that you can see here, and the other is the fibrillary tangles inside the neurons. So you can visualize here the neurons that are progressively this tau pathology is happening in the interior. So it is possible and necessary to integrate the studies of the brain from multiple scales. So and it's important and critical because beta amyloid and tau can affect neural activity at multiple levels of complexity, as some people today have emphasized, from the level of molecules, synapse, neurons, circuits, and network. But now it's possible not only because the computational method that you have been talking today, in particular about the network activity, but also at the level of the microanatomy, as I was explaining. So it's important to emphasize that in Alzheimer's disease, there's a progressive neurofibrillary and amyloid deposit in Alzheimer's disease. So you can see here that from tau, from the protein tau, there is the beginning is in the transcensorenal cortex, then the limbic structures, and then in the isocortical area, where with beta amyloid, the pattern of distribution is different. Just before is in the visual cortex and the temporal cortex, and then in other brain areas. So, and this is a correlation between the progressive loss of memory and the deterioration of behavioral and cognitive function with this progression of the disease. This is a very nice representation of William Molen, the self-portraits that are thought that he developed Alzheimer, and these are the different ways to how he interpreted himself self-portraits during the progression of the disease. That this is just only a metaphor to explain this. So one of the most interesting things from my point of view, because I am an anatomist, is that for many years, both Alzheimer, the tau and beta amyloid are asymptomatic and impossible to distinguish. Then you have the mild cognitive impairment with markers you can visualize. Then you have dementia. But there are many years that you have these pathological findings, but you are not demented. You don't have apparently any cognitive problems. So my question, the question that we are from a clinical point of view, is what alterations in the synaptic circuitry explain the early cognitive decline in Alzheimer's disease, and how can we prevent these changes? Why is it so hard to answer this question? As I said before, we know very little about the human brain, the structure of the brain, at the microanatomical level, but based on these new methodologies that we can apply to the study of the brain, now I think we can be optimistic. This is a way that we analyze what is the effect of beta amyloid in the brain. This is a staining with vesicular glutamate transporter and GABA in green, and GABA transporter for inhibitory terminals in red. And then this is a movie that where you can see that there are this is the neuropil, and this is, you can see the neurons are surrounded by both kind of terminals. So what happens when you have plaques in the brain, the deposit of the brain, then there is a aleatory deposit of plaques in the brain. So, and this is, you have to consider what happened with the plaques regarding to the pyramidal neuron. You have this a selective innervation of different types of interneurons. There are interneurons that make connection mostly with the dendrites, others are with the dendrites and soma, others are the basket cells that innervate the soma, and there are also cellular cells that innervate only the axon initial segment. All of them are inhibitory, and the functional is very different from each other. This perisomatic innervation that is gives rise to it's called perisomatic inhibition, which is crucial for the control of the activity of pyramidal neurons. And in this control, there are two major types: basket cells and cellular cells. The other type of dendritic cells are involved in plastic processing and other type of control of the activity of the pyramidal neurons, but not in the output of the neuron, because they are very far away from the axon initial segment initiation that is in the axon initial segment. But in the axon initial segment, you have these cellular cells that block the activity like this, no, in the brain. So what happens when you have in Alzheimer's disease? What happens is that there is a deposit of plaques, and we need to know what is the influence of this deposit in the family. My God, I will start. I will finish in my introduction. So you have what happened to this. No, so you have, for example, you have the deposit here. Then it's different if you have the deposit of beta amyloid in the cell body or near the cell body. And this is, for example, here you can see the combination of beta amyloid with the staining with vesicular glutamate transporter. You can see here in green are the neurons, and in red are the glutamatergic innervation. You can see that there's the soma, the neurons that are in contact with the soma, like here, there is a decrease in the number of GABAergic input. So we also apply fixed-end electron microscopy, and we found that there is about 80% of the decrease of the synapse around the plaques. So now I'm going to go very fast just to explain what happened with the regarding the dendrites. So what we are doing is just to make injections in neurons, just to visualize what happened with the dendrites when they touch the plaques. No? And here you can see it is an example that we inject the neurons, and we can see here the one neuron that is, in this case, we stain for tau. And when we combine the intracellular injection with tau, we can see that this neuron that was here, this one is tau negative. So we can compare tau negative, that means control neurons, with neurons that they have tau in a rectangular stage, because neurons, when they start to be damaged, before start with a rectangle and then a rectangular stage. And when there's rectangle, there are no changes in the number of spines. But when you have tangles, like in this case here, you can see that there is a decrease in the number of spines very clear. And this is something that I will come back in a moment. But in this case, you can see here a plaque. If we go higher magnification, you can see here that the plaque that is in contact with the dendrite, there are a decrease in the number of spines. You don't need to make an estimation because it's obvious here the decrease of the spines. This is another very nice example. You can see here, this is the dendrite with full of spines, and when you go near the plaque, you see it's like a very short segment that disappears the spine. So what they are doing, So this is again, and this neuron has been injected, and this is this is the tau positive neuron with tangles. You can see the spines of this neuron, that is the one, that is, there is no spines, and the dendrites are very atrophic. So these neurons are disconnected. So the accumulation of diffuse phosphotau in a rectangular state doesn't change the number of the spines, but when you have in a rectangular state, then the neurons start to be atrophy and loss of spines. So beta amyloid plaques induce local alterations, analytical somata in contact with beta amyloid, and this induces a loss of local dendritic spines and loss of perisomatic abiotic terminal. And this will give rise to hyperactive neurons, epileptiform activity. So because there are many, and then every time that you touch the neuron, the cell body disappears locally. This so the presence of tau led to a progressive dendritic atrophy and the loss of dendritic spines, and so it's a synaptic disconnection of pyramidal neurons, and this is probably more related with cognitive deterioration. Plaques will be just to give rise to this epileptiform activity typical of the Alzheimer's patients, but the loss of cognition and memory is related with pyramidal neurons. This loss of spines, progressive loss of spines. So, this in this neural excitation, inhibition imbalance is the endpoint of all Alzheimer's disease microarray synaptic pathology. You have the beta amyloid and the tau, and both of them give rise to an imbalance between GABA and glutamate, and the starting point to the network dilution in cognitive impairment. So it's something that we should include. So this is from a paper that we have recently published with my So now we can integrate the microscale, mesoscale, and macro scale from different levels, and creating a framework to test hypotheses and propose new strategies and intervention. So now today that have been you are talking mostly about this modeling of the whole brain, and this is a gap between the microscale and mesoscale, but I think that now it's feasible because, and one thing that will be fantastic is to go directly to the hippocampus, because the human hippocampus, we have already a lot of information, as I explained you, about the synaptic levels and about connections, and we know also very good in detail all the connectivity of the human brain, because it's very similar to the macaque monkey, and also there are also studies in the human. So we can now make a very precise model of the hippocampal formation. And this is important because, as I said before, in the early stage of Alzheimer's is the hippocampal formation, the sensorinal cortex and entorhinal cortex, the first regions of the brain that start to be deteriorated. And not only that, but also in epilepsy. In epilepsy, one of the most typical alterations in the temporal lobe of epilepsy is hippocampal sclerosis. And you can see here, it's amazing that you lose C1, the C4 here. So it's unbelievable how these patients with this damage of these circuits that are disappear, what is the influence on the activity. So the good thing, I believe, is that we have now the framework to work in a very detailed models of the human based on realistic data. Because now, for example, before when we made a model of a pyramidal neuron or synapses, number of synapses and so on, is based on extrapolation of data from animals. But now we are using humans, and the humans can be analyzed in a very good detail. It's not only the hippocampal formation that we are analyzing also many other cortical areas. The problem is that we don't have enough time to do these studies in other areas, but we have data about the morphology of pyramidal neurons in many other cortical areas. They are also about the synaptic proteins in the whole brain. They are also about the distribution of interneurons. These are a very interesting work, a microanatomical work of the human brain that usually is not included in these models. It's difficult, but would be fantastic to do it. And I think thank you. I'm sorry for this mess with the.
Sorry once again for the technical issues. We have a couple of minutes for questions.
A very provocative idea. I mean, you mentioned like beta amyloid. I mean, they're also known to well, generate or like not generate, but like control the gap junction proteins, like these connectome.